Tonight, you'll brush your teeth, climb into bed, and close your eyes. You won't think about the billions of cells inside you that are dividing, copying their DNA, and splitting in two. Most of the time, your body runs this process with perfect precision. But sometimes, deep inside a single cell, a molecular switch gets stuck. And that one stuck switch can change everything. You've probably never heard of KRAS. It sounds like a random string of letters, maybe a stock ticker or a license plate. But inside your cells, KRAS is a protein that acts as a binary switch. It's either off or it's on. When it's off, your cells divide only when they're supposed to. When it's on, it sends a signal, a molecular green light that tells the cell to multiply. And then it turns itself off again. That's how it's supposed to work. The problem is what happens when the switch breaks. In 1982, researchers identified KRAS as one of the first oncogenes, genes that have the potential to cause cancer. That discovery should have been the beginning of a straightforward story. Find the target, design a drug, save lives. But for 4 decades, KRAS resisted every attempt to drug it. The protein's structure was like a smooth sphere with no obvious crevices where a drug molecule could latch on. Scientists called these targets undruggable. The term was a white flag. After years of failed attempts, many researchers simply walked away. But a few didn't. Revolution Medicines, a California-based company, spent years working on a different approach. Instead of trying to jam the switch directly, they designed a drug that could trap KRAS in its inactive state. The drug is called daraxonrasib, and when the results from its clinical trials were presented at a cancer conference in Chicago, the room of thousands of scientists didn't just applaud. They stood up. They gave a standing ovation. You have to understand what that means. Scientists are not an excitable bunch. They deal in data, in confidence intervals, in the careful language of peer-reviewed studies. When they stand up and clap, something extraordinary has happened. The data that triggered that response was this. In patients with pancreatic cancer who had already tried chemotherapy, daraxonrasib almost doubled median survival time, from 6.7 months to 13.2 months. An extra 6 months of life, on average, for patients who had run out of options. Pancreatic cancer is one of the most aggressive forms of the disease. It usually causes no symptoms until it has already spread throughout the body. By the time most patients are diagnosed, their options are limited. Fewer than 10% survive longer than 5 years. The tumors are notoriously resistant to immunotherapy, a type of treatment that has transformed the outlook for many other cancers by unleashing the patient's own immune system to attack the tumor. The reason immunotherapy fails in pancreatic cancer comes down to that same molecular switch. When KRAS mutates, it doesn't just tell cells to multiply. It also remodels the entire environment around the tumor, creating a dense, hostile landscape that immune cells can't penetrate. The tumor builds its own fortress, and the body's defenses can't get in. Daraxonrasib changes that environment. By inhibiting KRAS, the drug doesn't just slow the cancer's growth. It may also make the tumor vulnerable to immunotherapy again. That's the theory. And if it proves correct in further trials, it could push survival times even higher. But here's where the story gets bigger than pancreatic cancer. KRAS is just one member of a family of proteins called RAS. Together, RAS mutations appear in about 20% of all cancers. That's roughly 3.4 million new cancer cases around the world every year. Colorectal cancer, lung cancer, endometrial cancer, small bowel cancer, stomach cancer, all of them can be driven by the same jammed molecular switch. Even multiple myeloma, a type of blood cancer, may respond to drugs that target RAS. Think about that number, 20%, 1 in every 5 cancers. For 40 years, that entire category of disease was considered untouchable by modern drug design. The proteins were too smooth, too cleverly designed by evolution to be easily disrupted. And then suddenly, they weren't. The reason scientists stood up in Chicago wasn't just about pancreatic cancer. It was about the sudden opening of a door that had been locked for 4 decades. You might wonder why it took so long. The answer lies in the physical shape of the KRAS protein. Most successful drugs work by fitting into a pocket or groove on the surface of a target protein, like a key sliding into a lock. But KRAS, in its normal state, is almost completely smooth. It has no obvious pockets. For years, medicinal chemists tried and failed to find a way in. The breakthrough came when researchers realized they didn't need to target the normal form of KRAS. They could target the mutant form, the version that's jammed in the on position. Mutant KRAS has a slightly different shape, and that difference creates a tiny vulnerability. Daraxonrasib was designed to exploit that vulnerability, slipping into a crevice that only exists on the mutant protein and locking it in place. It's a bit like finding a way to disable a faulty light switch by gluing it in the off position. The switch still exists, it's still broken, but if you can keep it from clicking on, the signal to multiply never gets sent. The implications extend beyond cancer drugs you take as an adult. There's a childhood cancer called neuroblastoma, where a different mutation disables a gene that normally acts as a brake on RAS. The brake fails, RAS runs wild, and tumors form in the nervous system of young children. If drugs like daraxonrasib can inhibit RAS-driven cancers broadly, they might eventually help those children too. And then there's the question of resistance. Cancer is an evolutionary process. Tumor cells mutate, adapt, and find workarounds. A drug that works today may stop working a year from now as the cancer develops resistance. Daraxonrasib is not a cure. The clinical trial results are promising, but they're not a guarantee that every patient will benefit or that the benefit will last forever. But that's how cancer treatment has always advanced. Not through sudden miracles, but through incremental victories that stack up over time. A drug that buys 6 months today becomes, when combined with other treatments, a drug that buys a year. Then 2. Then maybe, eventually, it becomes part of a regimen that turns a terminal diagnosis into a manageable chronic condition. That's the pattern we've seen with other targeted cancer drugs. It's slow, it's uneven, and it's often disappointing in the short term. But over decades, it adds up. What makes the KRAS story different is the scale of the target. 20% of all cancers, 3.4 million cases a year. When you find a master switch that controls that much of the disease, and you figure out how to flip it, you haven't just developed a new drug. You've opened an entirely new category of treatment. Revolution Medicines isn't the only company working on this. Now that the undruggable label has been shattered, competitors are rushing in. More refined versions of daraxonrasib are already in development. Other drugs that target different RAS mutations are entering clinical trials. The next few years will likely see a wave of new treatments that all trace back to this same breakthrough.
You can see why those scientists in Chicago stood up.
They were applauding more than a clinical trial result. They were applauding the end of a 40-year stalemate. They were applauding the moment when a problem that had been declared impossible was suddenly, demonstrably possible. And if you look at the history of cancer research, that's often how it works. A long period of frustration, a sudden insight, and then a rush of progress that had been waiting for the right key to turn in the lock. The standing ovation wasn't just for the patients who might live 6 months longer. It was for the possibility that, for millions of people yet to be diagnosed, the master switch might one day be something we know how to turn off. Tonight, when you brush your teeth and climb into bed, you still won't think about the billions of cells dividing inside you. But now you know that deep inside each one, molecular switches are clicking on and off with metronomic precision. And for the first time in 40 years, we're learning how to fix the ones that break.